In this video tutorial, we will go through the whole Mialab workflow for interactive COVID-19 lesion segmentation. You can load an input CT scan by clicking on Open Dataset and then Open Nifty File. We select one example image which will be loaded into Mialab, as you can see here. After that, we can select Workflow and COVID-19 segmentation. The first automatic lung segmentation step will then start running. When the automatic segmentation process ends, you can see your preliminary results in green. You can scroll through different slices and correct any possible errors by using the manual editing tools, such as the magnet tool that you see being used here. If the surface to be corrected is relatively wide, you can also adjust the sigma of the Gaussian function accordingly. In general, though, what is most important is to focus particularly on the lung regions that are affected by lesions and make sure that the lung segmentation is correct there. Here, you can see an example where the lung segmentation had to be slightly corrected in order to properly include all lesions. You can keep scrolling through all slices and correct as much as you wish and need. Once you're happy with your corrected lung segmentation, you can move on to the next step by clicking on Finish Manual Editing. This will prompt you to the threshold-based level set segmentation of COVID-19 lesions. Let the program run until you get a preliminary segmentation result, which is going to be indicated in green. You can scroll through different slices to check whether this preliminary segmentation is satisfactory. You can also change the segmentation mask opacity in order to better view the lung regions that are indicated as lesions by the software. In this case, we can notice that the brightest areas of these lesions were not included in the segmentation. This issue can easily be solved by increasing the upper threshold of the level set segmentation on the left side of the screen. Another possible issue is that the software may segment areas that do not necessarily correspond to lesions. To solve this problem, you can click on Allow Blocker Editing and then add a blocking sphere in the image. This will prevent the segmentation algorithm from including anything that is inside that sphere. The segmentation result can then be updated by clicking on Start. Another option that can be changed is the smoothing factor, which determines the degree of smoothness of the level set curve. This variable can be increased if you want to obtain smoother lesion boundaries. Remember to click Start after any updates in the parameters. Once you're quite happy with your preliminary lesion segmentation, you can click on Next Step. Here, you can have a better view of the segmented lesions, including a 3D rendering tool. You can then manually correct your segmentation in a very similar way to what was previously done for the lungs. For example, you may want to reduce the segmented area. This adjustment can be made again by using the magnet tool, or it could also be useful to switch to another tool, such as the brush. By pressing Alt on your keyboard, it is possible to use the brush tool to delete regions from the segmentation mask. Here, instead, the brush is used to add a small lesion that had not been previously segmented during the automatic segmentation step. You can keep performing manual corrections until you are happy with your COVID-19 lesion segmentation. Once you're done, you can save your segmentation mask by clicking on Export Mask. And that's it. This is the end of this Mialab workflow, and we hope this tool is going to be useful for your work.